Hey everyone, and welcome. Have you ever really stopped to think about the secret chemical story that's happening inside the food you eat? Well, today we are diving deep into the molecular logic of life, and we're starting with one of the most fundamental ingredients on your plate, carbohydrates.
Now, I know what you're thinking. That sounds like a trick question, right? But I promise you it's not. They seem totally different. One's food, the other's fabric you wear. But by the end of this, you are going to see how they are shockingly similar and how just one tiny chemical twist makes all the difference in the world. The answer is hiding in the amazing world of biochemistry.
And this quote just gets right to the heart of it all. See, life isn't some kind of magic. It's chemistry. It's this incredibly elegant organization of non-living stuff, just atoms and molecules, that builds everything from a single cell all the way up to you and me. So today, we're going to take a look at the rulebook for that organization.
And to really get a handle on that rulebook, we first need to meet the main characters in this story, the essential molecules that make all of life possible.
So, living things are basically built from four main classes of these big, complex biomolecules. You've got your proteins, you've got nucleic acids like DNA, and lipids, which you probably know as fats. But today, we are zeroing in on the group that provides both the fuel and the framework for almost all life on Earth, the carbohydrates.
All right, let's move on and really decode what a carbohydrate is. We're going to go beyond just what we see in the kitchen and step into the lab to see what they really are on a chemical level. We run into these things every single day. They're the sugar you stir into your coffee, the starch that makes bread so good, the sweetness in a perfectly ripe piece of fruit. All of these are carbohydrates. But what does that word even mean, chemically speaking? What's so cool is seeing how our understanding has evolved. Way back when, scientists noticed a pattern. A lot of these molecules had a formula that kind of looked like carbon atoms were just attached to water molecules, so they called them hydrates of carbon. Made sense, right? But it was a little too simple. The modern definition is way more precise. Now, it sounds complicated, optically active polyhydroxyaldehydes or ketones, but stick with me. All it really means is that these molecules have a bunch of alcohol groups plus one other special group, either an aldehyde or a ketone. That specific structure is the key to literally everything they do.
So how do we get from that chemical definition to the big, complex starches and fibers we see all around us? Well, it all starts with the basic building blocks.
Carbohydrates are basically sorted by size. And honestly, the best way to think about it is like Lego bricks. The smallest, single bricks, those are called monosaccharides. That's your glucose and fructose. Now, if you stick just a few of those together, you get oligosaccharides. Table sugar, or sucrose, is a perfect example. And when you link hundreds or even thousands of them into these massive chains, you get polysaccharides. That's the starch in your potato or the cellulose that makes up a tree.
And scientists have this really neat and logical system for naming these single bricks. It's based on just two things: how many carbon atoms it has and which functional group it's got, the aldehyde or the ketone. So a 6-carbon sugar with an aldehyde group, like our friend glucose, is called an aldohexose. See? It's a simple, descriptive name that tells you the molecule's core identity just by looking at it.
Okay, so let's zoom in on that aldohexose, glucose. It is probably the most abundant organic compound on the entire planet. And figuring out its true structure was like this brilliant piece of chemical detective work. So at first, all the clues pointed to a simple, straight-chain molecule. The detectives, the scientists, they started with its basic formula. Then, through a series of really clever tests, they proved that all 6 carbon atoms were linked up in a straight line. More experiments revealed it had 1 aldehyde group and 5 other alcohol groups. All the evidence pointed right here to this open chain structure. It seemed perfect. Case closed, right?
Well, not quite. The straight-chain model was good, but it just couldn't explain some really strange behaviors that chemists were seeing in the lab. There were some serious cracks in the theory.
So here were the problems that were driving them crazy. First, even though it supposedly had a free aldehyde group, it kept failing a classic chemical test for aldehydes. That made no sense. Weirder still, when they modified its 5 alcohol groups, the aldehyde group seemed to just vanish. And to top it all off, scientists figured out you could crystallize glucose into 2 totally different forms, which they named alpha and beta. The straight-chain model had zero explanation for any of this.
And then came the solution. And it was so beautiful and elegant. The molecule actually bends back on itself. The alcohol group on the 5th carbon attacks the aldehyde group on the 1st carbon, and the whole thing just snaps shut into a stable 6-membered ring. And this one change explained everything all at once.
The aldehyde group isn't really free anymore because it's locked into this ring. And what about those two different forms, alpha and beta? Well, when that ring closes, the new hydroxyl group on the very first carbon can point either down or up. And those two possibilities create the alpha and beta forms. There's a special name for them, anomers. So the big takeaway is that this one simple act of forming a ring solved all the mysteries of glucose's weird behavior.
Okay, so now that we understand our fundamental Lego brick, that glucose ring, let's move on and see how these bricks build up the massive structures we talked about at the beginning.
And here we are. This is it, the answer to our opening question about the potato and the cotton shirt. Both starch and cellulose are just long chains of glucose. The only difference is the type of linkage between them. Starch is a chain of alpha glucose units. Cellulose is a chain of beta glucose units. That one tiny change, just the orientation of a single bond, completely transforms the molecule's entire personality. The alpha linkages make a coiled shape, which is perfect for packing away energy. The beta linkages make straight, rigid fibers, which are perfect for building strong structures.
So let's bring this all together and just appreciate the massive impact these incredible molecules have on our world and inside our own bodies.
Their roles are, and I'm not exaggerating, absolutely vital. They are a huge part of our diet, giving us the energy we run on. Plants store that exact same energy in the form of starch. We animals store it in a slightly different, more branched form called glycogen. And of course, the entire structure of the plant kingdom, from a blade of grass to a giant redwood tree, is built from cellulose. So if you take away just one thing today, please let it be this. The simple flip of a single chemical bond, that tiny difference between alpha and beta glucose, is what determines whether a chain of sugar becomes an easily digestible source of energy or an incredibly strong building material. It is the fundamental difference between food and fiber.
Which brings us right back to the final answer. Why can our bodies break down those alpha linkages in a starchy potato, but not the beta linkages in a cotton shirt or a piece of paper? It's because our digestive enzymes are physically shaped to fit one, but not the other. It's just a powerful reminder that in the world of biochemistry, shape is absolutely everything. Life operates on an incredibly precise and really beautiful chemical code. Thanks for digging into this with me.